Okay, continue with paper 3. So what we're going to do here is we're going to do the question that we didn't do it in class. We will do maybe like 1 or 2 questions or maybe 3 questions. So there are 3 things over here. Number 1, it could be the voltage of the cell of the battery that affects the rate of photosynthesis. Number 2, it could be distance of light intensity that affects the rate of photosynthesis. Number 3, it could be concentration of carbon dioxide, concentration of sodium hydrogen carbonate solution that affects the rate of photosynthesis. So there are 3 experiments that you have to know, just in case whichever came out lah. So we're going to do the first question here first. The first question here is from this year's state paper, from Pahang state paper. I know a lot of school teachers did this question with you already, but it's okay. We will do it because I never do it in class, so I'm going to do it with you, yeah? Okay, number 3. Just now, many people asked me, what is the function of sodium hydrogen carbonate solution? This solution is used to provide carbon dioxide to the plant. Okay, it's used to provide carbon dioxide to the plant. And one more thing is sulfuric acid. Sulfuric, if there is a sulfuric acid, is to maintain the solution of the plant. Because if you add with sodium hydrogen carbonate is alkaline, when you add with a little bit of sulfuric acid, they are able to increase the rate of photosynthesis. They are able to provide an optimum medium for the plants to carry out photosynthesis. Okay, your setup should look something like that. The first question here is the voltage of batteries that affects the rate of photosynthesis. It says, green plants need to water, light, and carbon dioxide to carry out photosynthesis. As a biology student, you are required to conduct an experiment to study the effect of light intensity on the rate of photosynthesis. Okay, it says this is the light intensity rate of photosynthesis. Usually in the question, you can find your MV and RV. Usually your manipulated variable and responding variable. Light intensity is your manipulated variable. Rate of photosynthesis is your responding variable. Okay, this is your setup. So you put your solution inside the boiling tube, and then you clamp it, and then you set up your light first. Before you put your hydrella, maybe you should learn how to set up the apparatus first. Okay, remember to connect your input and output of a battery correctly. Okay, then we have to put a thermometer to make sure that the solution is constant temperature, in room temperature. Anyhow, it's constant lah, guys, because to put or not to put a thermometer, it's not a big deal over here, but just take it lah, just put a thermometer lah, and you put your hydrella by using a forceps. You clip the hydrella, you put a paper clip. The paper clip is to ensure that the hydrella species don't float. You have to submerge into the water. You have to make sure that the entire species is in the water itself, in the solution itself, in the sodium hydrogen carbonate solution. This is to ensure that the plant able to produce enough of gases out. Okay, and then your light has to face towards the plant. The distance, it has to start from the plant, usually from here lah. You have to start from the boiling tube also can, from the plant also can, from the boiling tube, all the way to your light source, the light bulb, not the retostat, the light bulb. This is your distance, about 20 cm. So you're going to put it like that. Okay, and then usually when you on your light, you have to let them to adapt to the situation. So let's say it's 5 minutes, right? Maybe the first minutes, you have to let the plant to adapt first. If you see the bubble coming out already, then you have to count your bubbles. But usually they would let maybe 1 to 2 minutes for the plant to adapt to the situation. So if it's 5 minutes, maybe the first minute, you let the plant to adapt. But you have to observe the plant. Okay, try not to spend too much time on doing experiment, because if you spend about 30 minutes to do experiment, you left only 15 minutes. I don't think it's enough for you to answer your question. Maybe you should go to your question to see the question first. See which one that you can do first. For example, maybe you can write your MV, your RV. Maybe you can start to draft your graph while you're doing the experiment. Maybe you should learn how to write your operational definition. These are the things that you can answer first. Without doing the experiment, you have your answer, because you know this question going to come out in exam, right? So you write the answer first. Later, if it's not enough time, right, you have no marks. Doing experiment is not the thing your teacher will see. Your thing the teacher will see is your paper. Your paper, you have to finish your paper, guys. Okay, the procedure here says that based on diagram 1, carry out the experiment by using the step below. Attach a paper clip under the stem cutting hydrella species. When you cut the stem, you put a paper clip. Why? Because as I mentioned, you have to allow them to sink into the water and put them into boiling tube containing 0.2 sodium hydrogen carbonate solution. Okay, it will be your constant variable. Okay, this is your constant. This is your constant variable, constant variable. Clamp the boiling tube vertically to the retort stand. Light the 60 watt, typing error, 60 watt bulb with a distance of 20 cm, 20 cm from the hydrolyzed species by using one battery, 1.5 voltage. So 20 cm is also a constant variable. This one, later you will know, this is your manipulated variables. Okay, after 5 minutes, 5 minutes is also your constant variable. Count and record the number of air bubble release for every 2 minutes for 6 minutes. Means every 6 minutes, within the 6 minutes, every 2 minutes, you're going to count. But when you see there is bubble, then you count, because sometimes the bubble will come out before 2 minutes. So just count when you see a bubble, then you count. When you count your bubble, it's supposed to count when they float on the surface. When they stick to the leaf and it's big and they stick to the leaf, then you just count lah, okay? So when they float, you count. So this here, number of bubble release, it will be your responding variable, RV, responding variables. Replace the 0.2 sodium hydrogen carbonate solution in the boiling tube with a new one. So you're going to pour it out. You're going to replace a species again, because this is how you ensure your experiment is done accurately. Because if you reuse it, maybe your result is not accurate. So you make sure that you replace it lah, okay? And then you repeat step 3 and 4 by using 3 battery. So then you have your different manipulated variable because it's the different voltage, right? Record the data collected in table. Construct the table and record the data collected from this experiment. The following title should appear on your table. So they will give you this and you have to draw this in your table. The formula, usually they will give you an exam, so don't have to memorize the formula, but the formula here is number of gas bubble release divided by time. It's always the same formula over here. When you draw your table, remember to label on top. This is voltage of cell. Remember your unit is V. Number of air bubble release in 2 minutes. Then the unit here should be minute. Okay. Average should be minute. Okay. And then your number of air bubble release by time, it will be minute minus 1. Okay. This is your unit. When you're drawing a table, unit is important as well. Your answer, if it's not the same as your teacher, is okay, because there's a plus minus. Maybe plus 1, 2, or maybe 2, 3. So if your answer is not like that, it's okay, guys, because it's not always the same answer that you will get. Because just now we say, right, 6 minutes, right? At every 2 minutes, they will release 2 bubbles. 2 bubbles, 2 bubbles. In total, we have 6 minutes. Within 6 minutes, they have 2 bubbles being released. And then we have what? At 6 minutes, we change the voltage to 4.5. They release 777. I mean, if the question that if you are doing the experiment, if you don't know the answer, just fake your answer. You have no choice, just fake your answer. Then you have your average, and you have your rate of photosynthesis. You use number of bubble release, divide by time. What is your time just now? Your time is 6 minutes, so you divide by 6, yeah? 6 minutes, okay? Then we have your answer. Okay, next. Based on your experiment, conduct state 1 observation. When you write about observation, you need to write MV value, RV value. You need to include your value in your observation. If you forgot the answering technique, remember, go back to my previous YouTube video and watch the answering technique, okay? You're going to write MV value, RV value, okay? MV value, RV value. You can choose the highest or the lowest, whichever you prefer. You want to choose the highest also can, lowest also can. So you're going to say, if the voltage of dry cell is 1.5, okay? 1.5 is the value, right? This is your MV, okay? If your MV is 1.5, the number of bubble release in 6 minutes is your RV, is 6. This is also your value. But depending on your answer, depending on your result. But if it's 1.5, then it will be 6. Or you want to write the other one also can. Just choose to write whichever one. You want to write the highest one also can, the lowest one also can. Remember, observation, you have to write value. MV value, RV value. Okay, next, state 1 inference based on any observation. Inference, no need to write value. You choose the highest or the lowest to explain. Just now we write what? The 6, right? Then you can choose to write 6. If it's not, you can choose to write this also can, whichever. Doesn't matter one, both also correct, but I would suggest you to follow back whatever you choose from your hypothesis. Not hypothesis, your observation. Choose from your observation. So you're going to say, the number of air bubble release increases due to higher light intensity result in higher rate of photosynthesis. This is based on the second answer, when you have more light. If there is lesser light, what you're going to write? You're going to change the increase to decrease. Change the higher light intensity to lower light intensity, higher rate of photosynthesis to lower rate of photosynthesis. So what is involved in your answer? You need to write your RV, okay, and then your MV, which one higher, lower MV, and then your ORV, rate of photosynthesis is ORV. What does ORV mean? Operational responding variable is the one that you learn it in your book. Number of air bubble release is what you see in your experiment. Both are responding variables. Usually when we write, we will always choose to write responding variable first before we choose operational responding variable. State the method to handle the variable based on experiment. Method to handle. We also have answering technique for method to handle. Okay, if you want to write responding variables, because if you write number of bubble release, okay, these are number of bubble release, your keyword of you have to write is you're going to write count and record, okay? You need to write count and record, that is the keyword. Count and record the number of air bubble release in 6 minutes by what? Observation, because we have no apparatus here. When there is apparatus, we have to write apparatus, but this one got no apparatus. So count and record is the keyword. Count and record your RV. Count and record your RV, the number of air bubble release in 6 minutes by using the apparatus. If you don't want to write this, you can write rate of photosynthesis. Your ORV can be rate of photosynthesis. Rate of photosynthesis. If it's rate of photosynthesis, we have to write calculate and record because we have a formula. You need to write the formula if you want to write rate of photosynthesis. Both answer is correct, guys. If you want to write responding, you have to write count and record. If you want to write rate of photosynthesis, you have to say calculate and record with the formula given. Number of bubble release divided by time. Remember, if they don't give you formula, you have to know the formula by yourself. It's only one formula that you have to remember. Okay, next, the constant variable. You can write power of bulb, concentration of sodium hydroxide carbonate, or the species. So if you want to write power of bulb, your keyword here is fix. Fix the power of bulb, your constant variable, at 60 watt. 60 watt, that is a watt that they give it to you, 60 watt, right? 60 watt, okay? Next, concentration of hydrogen, sodium hydrogen carbonate. Same thing, you have to write the word fix. Fix what? Concentration of sodium hydrogen carbonate, constant variable, at 0.2%. Because we have no apparatus for concentration. So you're going to write fix the concentration at 0.2%. Hydrilla species, fix the type and size of the hydrilla species. Did they cut the hydrilla here? Forgot already, guys. Did they cut the hydrilla? They didn't write the size of hydrilla. Sometimes if they cut the hydrilla, they will write the size. So you can say, fix the size of hydrilla by using a ruler if you want to write hydrilla species. So remember, the keyword for responding variable is count and record. The keyword for constant is fix the constant at what value by using what. Okay, next, state the hypothesis of this experiment. Because in photosynthesis, we can write the higher, the what, the lower, the what. MV, we have a value. RV, we have a value. Both, we have value. We can write the higher, the lower. Okay, the higher, the what, the lower, the what. Then we can say, as the voltage of light increases, voltage of light is your MV, increases. Okay, the number of air bubble release, this is your RV, increases. Can you see? The higher the MV, the higher the RV. You can say the higher the light intensity, the higher the rate of photosynthesis. Both also correct. Okay, you can write the higher the light intensity, the higher the rate of photosynthesis. Hypothesis, both answers are correct. Next, they ask you draw a graph of rate of photosynthesis against dry cell voltage using the graph paper provided. Okay, now why is this question a bar chart? Because the dry cell voltage is a constant number. It's 1.5 and 4.5, and there's only 2. Because if you draw a graph and there's 2 dots, right, it's a very weird number. So they will draw a bar chart when you talk about dry cell voltage. So make sure that your graph, you have to label your XY axis. Remember that at the bottom, you have to write battery of power V, and this is rate of photosynthesis, minute minus 1. You have to make sure that your graph is 70% of your graph paper. The title you want to write, you can write, but if you don't want to write, it's okay. You can say rate of photosynthesis, the graph against the battery power, or battery power against the rate of photosynthesis. To make sure that you write, okay, you have to make sure that you have axis, XY, two correct point transfer, which is your answer here, and uniform scale. All graph is the same, bar chart is the same, but this one they use a bar chart. Usually we use a graph because we don't use dry cell, we use the distance, right? Distance cannot draw a bar chart, we have to use a graph, yeah? Based on the graph in E1, state the relationship between rate of photosynthesis and the light intensity. The question says what? Based on the graph, rate of photosynthesis and the light intensity. You cannot write more bubble release because it's not related. You have to write back to what they want in the question. So the higher the light intensity, the higher the rate of photosynthesis. Actually, the answering technique is very similar to hypothesis, right? But this one, you have to write rate of photosynthesis. Hypothesis is the higher the MV, the higher the RV. This one, the higher the OMV, the higher the ORV. Just follow the question. Just write the answer according to the question. It's a question that you can do it by yourself. So that's why I say, before you do experiment, there are some questions that you can do it already. You don't know, higher light intensity, higher rate of photosynthesis. Of course, you know what. So this one don't have to do experiment. You also know the answer. So you can try to do first, because doing the paper is more important than doing the experiment. Okay, next. Okay, explain your answer. How do we explain our answer? Our explanation need to base on biology knowledge, because the question here, they say state. The next question they ask you to explain, right? How do we explain? We have to write. Because when there is more light intensity, we have higher light-dependent reaction also. When you have more light-dependent reaction, more oxygen is being produced. Okay, the keyword is more. More oxygen is being produced because we have higher light intensity, higher light-dependent reaction. Then this is your answer. Your explanation needs to base on a little bit of your theory. Okay, or you can write the other one. Lesser oxygen release when the dry cell voltage is lesser, it shows a lesser light intensity, lower light intensity also can. So whichever one, slash, it means either one correct also can. So this is about your voltage of cell affecting your rate of photosynthesis. Okay, and then we did this in class also just now. The graph here shows this one, right? Just now we didn't draw a graph, but the graph is like that. When you join the point, remember the first to the last point, use a free hand to draw. Don't use a ruler, because ruler is a bit weird, because this is a printed one. That's why they never use a free hand. It's supposed to be a free hand. You draw the graph. When you draw a graph, remember, don't pass through this line. Means when you draw a graph, you cannot draw over here and over here. This is a wrong graph. You need to make sure that it's first to the last point. Don't need to start from the like zero. First to the last point, yeah, remember to freehand and draw your graph. Okay, and then this we did just now. And then question 3. Just now we did halfway, right? Okay, this question here is the effect of carbon dioxide concentration on the rate of photosynthesis of Hydrilla species plant. This is your setup that you can see. Sometimes in your light bulb, they will put a paper cup on the outside. If your teacher give it to you already, if your teacher set up for you, then it's completely fine. Because in one of the apparatus, maybe they have paper cup or maybe plastic cup lah. They'll put it on the outside here, and then you have to make sure that the light has to face towards your plant. Paper clip, if there is a plasticine without the retort stand, you can put a plasticine at the bottle to make sure that you can make sure that the boiling tube don't move lah. If there's a plasticine or modeling clay. But if your teacher set up the light for you, if guys, I don't think there's so many if lah, but if the teacher set up for you, you just have to put a battery, set it up. Okay, we will not do this question because I did half right. What I want you to know here is in your exam, if you cannot really find your result, you just fake your result. If you don't have your result, you fake your result. How do you fake your result? Because the result here given to you, it was 0.1%, 6 bubble is being released. 0.2% is 8. 0.3 is 12. 0.4 is 20. 0.5 is 24. If it's 5 minutes, you use 6 divide 7 times 5. Fake a bit lah, guys. Plus minus never mind one. But your result, it will not be 50, right? If your graph is like that, imagine that your graph looks like that. One here, one here, one here. Of course, your answer is wrong. Cannot be like that, what? So you fake your answer to make sure that your line is smooth a little bit. It's slightly smooth. Either it's like that or like that. Cannot be like that one, guys. Not possible. Because it's the concentration that is supposed to increase or decrease gradually. It's not supposed to do like a roller coaster. Okay, not supposed to be like that. And the rate of photosynthesis, the calculation, they use number of gas bubble release divided by time. Time is 7 minutes, right? Just now we stop here, right? Based on the result in table 1, calculate the rate of photosynthesis for each concentration of sodium hydrogen carbonate solution using the formula given. You use 6 divide by time, you get this answer. Okay, you use 8 divide by time, you get this answer. And then we have 12 divide by time, you get this answer, and so on. You will notice that rate of photosynthesis, you can have a decimal place. Your decimal has to be constant, means 2 decimal means 2 decimal for everything. 2 decimal is your maximum because if you write 3 decimal, it's going to be very hard for you to draw a graph. So fix your decimal into 2 decimal for this experiment. So 1.71, 2.8, okay, it's completely fine because you have to use this to draw a graph. Most of the time, if the question wants you to draw a graph, you have to draw a graph. Okay, I ask you, based on the result in C, explain the relationship between the concentration of sodium hydrogen carbonate solution with the rate of photosynthesis. They ask you to do what? Concentration of sodium hydrogen carbonate, rate of photosynthesis. This is your MV. This is your ORV, operational responding variables. The higher the concentration, just now we say, right, what is the function of this thing? To provide carbon dioxide. The higher the concentration of sodium hydrogen carbonate, the higher the rate of photosynthesis. Your answer need to base on what they ask. Please don't write higher the concentration of sodium hydrogen carbonate, more gas bubble is being released, not related. Write rate of photosynthesis, or you can write the lower, then the lower the rate of photosynthesis. We don't write vice versa in exam. This is just for your marking scheme purposes. How do we explain this? Because when we have more concentration of sodium hydrogen carbonate, we have more carbon dioxide release. Just now we say, right, more carbon dioxide release, more oxygen is released. Don't have to talk about glucose because it's not quite related. Just write more oxygen is released. Predict the rate of photosynthesis when the distance between the bulb and Hydrilla plant is increases. Increases means what? Further away. Further means what? Of course, the rate of photosynthesis, guys, they ask you rate of photosynthesis. Of course, decreases because the further away means lesser light is being received. Lower light intensity, lower oxygen gas produced, lesser oxygen gas produced. This is your answer. Lower rate of photosynthesis, lesser oxygen release. Okay, so this is your answer for carbon dioxide. Then, wait, let me find another question that I want to do. Yeah, this one, okay, let's do this question. You have to carry out an experiment to study the effect of light intensity on the rate of photosynthesis. Diagram 1 shows the apparatus setup of this experiment. A 60-watt bulb is used as a light source and temperature of 0.2% sodium hydrogen carbonate is 28 degrees Celsius. So they give you temperature 28, they give you 60-watt bulb, and they give you 0.2 sodium hydrogen carbonate. All of these are constant. What is your manipulated variable and responding variable? Light intensity, rate of photosynthesis. This is not about light bulb, this is about distance. So this is your manipulated variable. This is your manipulated variable. This is your responding variable, operational responding variable. Because rate of photosynthesis is the one we learn in textbook. Your responding variable is supposed to be bubble release. Okay, this is another setup. I don't think you will do this because there is no flux is providing in your apparatus and material. So you follow the question just now, the one I showed you just now, that will be your setup lah. And then we have a ruler. Just now I told you how to measure already, right? Plan your experiment using apparatus and material. Your procedure should include method to handle, precautionary step. As we have mentioned, biology has high chances of guided experiment. Hopefully it's guided, but if it's unguided, these are the questions that you will see in your exam. If they ask you to write procedure, this is how you're supposed to write your procedure with the method to handle. Okay, we have method to handle for your MV, method to handle of your RV and your CV. Remember, for responding variable, for manipulated variable, and your constant variable, we have a keyword. For manipulated variable, your keyword is use different. Use different distance of light source. Okay, this is your manipulated variable with Hydrilla species, these are the value. You have to write your value when we talk about method to handle. If it's a responding variable, you're going to write observe and record, but better to write count and record because you are counting the bubble, right? Count and record number of bubble release in 5 minutes. Okay, this is your responding variable. Because there's no apparatus, you can say by observation. By observation. Or you can say calculate the average number of air bubble release in 5 minutes. You're going to write by using formula. The formula is number of bubble release divided by time. You're going to write the formula out. If it's a constant variable, you have to write fix your constant of sodium hydrogen carbonate. This is your CV at whatever value. Okay, and then you're going to write fix the temperature at 28 degrees Celsius. You can say by using thermometer. Okay, fix the size of Hydrilla species. Okay, if you have to cut the Hydrilla, you can cut 5 cm, 4 cm, doesn't matter lah, okay? At 5 cm by using ruler. You can say by using ruler and scalpel. Scalpel is the knife, yeah, it's called scalpel. Your precautionary step. In exam, if they don't ask you to write your procedure, sometimes they ask you to write your precautionary step. So remember, 3 guys, never mind, just only one experiment, just remember 3 is the same thing. You have to say, the Hydrilla species that is cut should always be outwards. It means you have to make sure that it's outward, but you have to put a clip, you have to make sure that it's in the water. Or you can say, Hydrilla species which is cut should always immerse in solution so that a lot of air bubble can be released and can be seen clearly. So you're going to put it into a solution, immerse fully also can. Every reading has to take 3 times to find your average reading. But because of time restriction, we cannot repeat 3 times because let's say 5 minutes, right? When you repeat, then you have no time already. So I would just suggest you to maybe, if they ask you to write the step, you can still write the same answer because you're not doing it, so it doesn't matter. And replace 0.2 hydrogen carbonate solution in the boiling tube with a new one. When you are doing a new experiment, you have to replace it because your sodium hydrogen carbonate solution is the one that provides oxygen. After you do 1 time already, then no more carbon dioxide. So it means your result is not accurate at the back. So you're going to replace. Your teacher should give you enough of solution for you to replace. Hydrilla, I'm not too sure. Because you cut, right? Cut 4 cm, maybe it's enough also. So you have to repeat your experiment when you are doing for another manipulated variables. These 3 things, you have to remember. If they ask you to write, you're going to write these 3 things, okay? If they ask you to write. Okay, next, carry out the experiment. State your observation on the number of bubble release in 5 minutes when the distance between the light source of Hydrilla is 60 cm. This is a prediction question. So they ask you to predict the question, prediction question. Observation means you have to write at 60 cm of light source with Hydrilla. This is your MV, your value. Okay, the number of air bubble release in 5 minutes. This is your RV. And then your value. Because this experiment, they repeated the experiment. So that's why they have first, second, and third value. But if you never repeat the experiment, you can just say the number of RV release in 5 minutes is whatever value. Because this is how many cm? 60 cm. It's in the MV also. Sorry, I thought it's a prediction. So 60 cm, you just write 60 cm one is whatever value. So this is your, literally is in the question. I thought it's not in the question, it's in the question. So just write the value. State the hypothesis of this experiment. When your MV has a value, you can write the higher the MV, the higher the RV. Or you can say the higher the OMV, the higher the ORV, both also correct. The higher the light intensity, MV, the higher the rate of photosynthesis, OMV, ORV, or you can write the nearer the light source to the plant, the more bubble release in 5 minutes. A bit weird, maybe I should say. The nearer the distance of light source with hydrilla species, the higher the number of air bubble released in 5 minutes. Okay, this one and this one also can be hypothesis as long as your result is correct. In hypothesis, there is no need to write any value one, just higher or higher lower, whichever one. State the constant variable in this experiment. Just choose either one to write. This one we highlighted a lot of constant, right? All of these are constant. Okay, what? Concentration, light intensity, the temperature. You can write all of these, whichever one. The temperature, the concentration, the voltage, and the type of size of hydrilla species. So this is the result. Because this experiment, there's no result in the book also, so there's no result. But for the result, it's also in the notes that I give it to you already. You can just follow the distance, just fake your result. If you cannot do it, I give you this result. For those who have no material, let me give you fake a result. Fake this result, okay? This is your result that you're supposed to fake. Your plus minus lah, okay? This plus minus answer. If it's more, then you add a little bit lah. This is your result that agak a bit lah, okay? This is your result for distance 5, 10, 15, 20, 25. You agak a bit, then you have your answer already. If you cannot get the answer, if you cannot get your answer, guys, if you cannot get your answer, if you can, you follow your own answer. Okay, next, classify the factors that affects rate of photosynthesis accordingly. We have internal factor, we have environmental factor. Internal means it's in the plants. So in the plants, we have what? Then the answer is given to you also lah. Thallicate mesophyll set is in the plant. Chloroplast is in the plant. Spongy mesophyll set is in the plant. Stoma is in the plant. This is other factor, outside factor. Environmental factor is outside factor, yeah. Classify is one of the questions. In the entire paper 3, we only have 11 type of question that you will see in your exam. Okay, next. Another group of pupils carry out same experiment where the distance between light source and hydrilla species is 20 cm, but using 0.2 sodium hydrogen carbonate with 60-degree temperature in beaker. Predict the number of air bubble released in 5 minutes. Okay, this question, only this is a prediction question. So prediction question, you can write more than or less than your result. At 60, for example, just now, at 20 cm, 0.2, let's say is, for example, 20 lah, okay, 20 bubbles lah, approximately, guys, actually just 20 lah, approximately only, okay. Let's say if it's 20, you're gonna say the air bubble release will be lesser than 20, okay, the number lesser or more than lesser because it's 60, right? When the temperature of 0.2 sodium hydrogen is 60 degrees Celsius compared to 28, this is your prediction. When they ask you to write prediction already, you have to explain your answer. We have to remember always to carry out photosynthesis, it requires photosynthetic enzyme. They have optimum temperature. High temperature causes the photosynthetic enzyme to denature. When they denature, photosynthesis cannot carry out. That's why lesser bubble will be released because the enzymes denature. Usually prediction question requires a little bit of your theory knowledge. So this is your distance. So we did distance, we did voltage, we did carbon dioxide also, the concentration also, right? So for my own student, you can try the rest of the question by yourself. I will give you the answer. If you have still any question after you read the answer, you can ask me. If it's not, then good luck for your exam, guys. Hopefully you will score 15 out of 15, okay? Okay, bye-bye, guys. Bye-bye.